Thank you. Good afternoon, everyone. I hope you are enjoying your day so far and the eloquent talk we had just now from Sarah. I'd like to thank the Macular Society for inviting me and giving me this opportunity to talk to you today. It's always nice to talk to real people. I usually spend my day locked in a laboratory talking to test tubes and students, and of those I'm not entirely sure was giving the most sensible answers. So it's nice to get out and actually meet people who are really affected by these conditions and get some feedback as to what's going on. I stand in front of you today feeling like a bit of a fraud. I'm not an ophthalmologist. I am an immunologist. So I'm interested in how the immune system is regulated around the body. And in fact, when I was a PhD student just up the road in Oxford, I'm almost ashamed to say I actually had no interest in the eye either. I was far more interested in how the immune system is regulated in systemic disease, so diseases that affect the whole body. But then in 2005, whilst I was drowning in a mountain of paperwork with a very unimpressed supervisor, somebody worked out that a series of mutations in a person's DNA could moderate their risk of developing AMD. And this was a big, big thing, certainly in terms of biochemistry, because when you had a target, something to study and look at, you were one ginormous leap closer to understanding how the disease progresses. Now, most of these mutations in your DNA occur in genes responsible for part of the immune system called the Complement Cascade, and you heard Sarah talking about that very, very briefly just now. I'm not going to bore you with the intricate details of what the Complement Cascade is. It's actually very, very complicated at its biochemical level, but at its very basic level, it's incredibly simple. It's a series of proteins, these molecules that float around your body, that are responsible for protecting you against bacteria, invading pathogens, and directing your adaptive immune system, your antibodies, to a target. But importantly, it's also important for removing unwanted immune complexes, cell debris, and protecting your surfaces and your cells from inappropriate immune attack, so autoimmunity essentially. And that's important to know because the biggest, or one, I should say, of the biggest genetic risk factors for developing AMD occurs in one particular gene, a gene called complement factor H. So what, you might think. But factor H is in fact the main regulator of the complement cascade. In fact, it is a blood-borne protein. It's all over your body at any one time. But it is responsible for protecting, in the eye, very importantly, Bruch's membrane. Again, you heard Sarah describe Bruch's membrane, this very thin, what we call acellular, so there's no cells in it, there's a very thin membrane that underlies your retinal pigment epithelium cells. And that's interesting, because drusen form inside Bruch's membrane. And we were very fortunate a number of years ago to find that this single mutation changed the way your immune system protected Bruch's membrane. And although it didn't give us a cure for AMD, certainly it gave us a far better idea and understanding that complement, although not necessarily initiating the disease, certainly aggravates it. So individuals who have these mutations develop the condition much earlier in life, much more virulently. This mutation, interestingly enough, is very prevalent. It's present in 30% of all people of European descent, and that's quite specific. And if you have this mutation, you're up to 3 times more likely to develop the disease. And you often wonder, or we've often wondered why it's so prevalent, why nature tends not to keep mutations in your DNA that are not useful. It's all about evolution. It's not designed that way. Why keep this mutation? Well, a very clever chap in America called Thomas Avery did a bit of research. He's a surgeon. He likes to dabble in research in his spare time. He's very, very good at it. And he worked out, if you have a map of the world and plot on the map of the world the incidence of this mutation in the population, so lots in Europe and America, very little in Asia, virtually none in Africa, and then overlay the incidence of age-related macular degeneration, sorry, they fit perfectly, but if you overlay that with the incidence of the Black Death in the Middle Ages, they actually map rather beautifully. And it turns out that this mutation in your factor H protein, the one protecting your body from its own immune attack, is actually recruited by a little bug called Yersinia pestis. Yersinia pestis, of course, is responsible for the Black Death, the bubonic plague. And it turns out that this mutation stops this little bug recruiting factor H from your blood and using it to protect itself from your immune system. So all these people in Europe who were being struck down with the Black Death, genetically and evolutionary, were being protected by this mutation. And unfortunately, for us, Mother Nature wants to keep us alive long enough to pass on our genes and live a healthy life up until our 40s and 50s, but after that it doesn't worry too much, and that is why there's such a high prevalence of this mutation in our DNA pool. But aside from that, we now have a target. We now know that an overactivation of your immune system is predisposing people to developing AMD. We have a target, we have something we can work with, which is very, very important, and there's an awful lot of work focusing on trying to get your immune system back on track. In fact, one of the drugs was mentioned as the anti-complement factor D, which is an excellent drug. We have very high hopes for it, in fact. It is in phase 2 clinical trials, but there's a long way to go before we know exactly what it does, but it does look very, very promising. And given the fact that the association between complement and AMD was only really made in 2005, we've come an awful long way very, very quickly. 10 years is not a long time to work out how a disease progresses and develop a therapy against it. So there should be a great deal of optimism that something will happen or come out of all this. There are, of course, other therapies. As Kathy sort of alluded to, there are stem cell therapies that are currently under investigation in various laboratories around the world. We've been very lucky to be supported by the Macular Society and by all of you with some of the research that goes on. And in fact, gene therapy is another potential therapy that is being trialed in various different laboratories. And some of you may have caught last night the One show on BBC One. Professor Macular from the eye hospital up in Oxford was on with a patient who suffered from another particular eye disease, not AMD. And they were injecting new genes, essentially, delivered by viruses into the back of this gentleman's eye with rather remarkable results. That sounds quite freaky, injecting a virus into your eye and hoping it replaces your genes, but it's not quite as archaic as that sounds. We've been using adenovirus delivery gene therapy in the laboratory for years. It's only just now being proved to be reasonably safe, and we're going forward into the actual clinic. Genetics tells us an awful lot about disease, but it's the biochemistry that will get you a cure. And the research that goes in laboratories is very, very important. Access to samples is particularly difficult, especially when it's something like human eye tissue. And that lady asked a rather astute question with regard to animal models. And although we don't use them in Manchester, they are used quite a bit. But it was recognized a number of years ago that this really isn't really good enough. And we needed to have access to the tissue that was being affected. And we're very fortunate in Manchester to have the Manchester Eye Bank, which is responsible for about 2,000 corneal transplantations. 2,000 pairs of donated eyes to come in for corneal transplantation. And up until January this year, when the surgeons removed the cornea for transplantation, the rest of the eye was getting thrown away, which we were horrified at. And so we contacted the Macular Society and said, this has got to stop. We need to do something about this. And they agreed, and they helped fund what is now known as the Manchester Eye Tissue Repository, which allows us to take the rest of the material after surgery, characterize it, we call it phenotyping, but characterize it for various different retinal dystrophies, extract DNA, and store it such that any researcher who wishes to look at curing eye disease can contact us and have access to this material that they would otherwise not have. And to give you an idea of the scale of this, it is only the second eye tissue repository in the world. So the Macular Society really has helped a great deal in setting that up. And it's in its infancy at the moment, but within a couple of years we will have however many thousand samples for people to use in research, and they'll can ask us and we'll be freely access to them. So we are very, very grateful for the support we've had. Something else that we're doing in Manchester, but there are other places in the country doing it also, is visual prosthesis. So this is when there's surgery to implant a chip into the back of the eye that allows an individual who has lost their vision to be able to see lights, shades of different shades of light, and so on again. And there was a very famous, certainly famous up in Manchester, case where a gentleman who had been blind for a number of years had this surgery, and he was wearing these special glasses which transmitted a signal to the chip in the back of his eye, which then distributed it up to his brain. And he was able to walk unaided from the front of the eye hospital down the car park and back again. And this man hadn't seen for about 20 years, and he was able to do it. And he could see different shades of light. He couldn't see particular objects, but with time and training, he's actually managed to find himself being able to read short words and short sentences again, which is quite phenomenal if you think about the fact he had completely lost his vision a number of decades ago. The great thing about my job is I go to work every day, and my remit is to do things that's never been done before. That's my job. And it's very nice when sometimes things go right. I do spend 90% of my time failing, by the way. 90% of the time we try something and it very much doesn't work. But when it does work, it's an amazing feeling. It's an amazing feeling that you can make a difference to people's lives, because we do know how much of an effect this has on every single one of you. You can rest assured that there are thousands of researchers up and down the country working every day, developing new techniques, developing sometimes outlandish and crazy ideas, but sometimes it's the crazy ideas that work. Sometimes following the norm with everyone else just means we all end up down the same dead end. So rest assured there are lots of people working on your behalf. And like I said, I think there is very good reason to be optimistic that in the future there will be treatments for the dry form of AMD. In fact, my laboratory, we are taking a subtly different approach in so much as we are looking at ways of preventing the disease ever happening in the first place, rather than treating the disease at the point at which it has started to take vision. I would much rather give somebody a drug or a treatment that means they won't go blind at all, or at least push it back far enough such that it doesn't become a problem within their lifetime, their natural lifetime. But whichever method prevails, we will continue, and with your support and help is what makes it possible. So I'm going to leave it there and leave plenty of opportunity for questions, please, and thank you.